Hello and welcome. If you're just joining, we are going to get started in another 2 or 3 minutes. Hello and welcome. If you're just joining, we'll get started in a couple of minutes. Welcome to everyone who just joined. We're going to give people a few more minutes to log on and then we'll get started. Thanks for joining, everyone. We'll give it about 1 more minute. All right, we'll get started. Hello, everyone, and thank you for joining us for this event. My name is Ann Johnson, and I'm a media liaison with Experimental Biology. This is a press conference for the Experimental Biology 2022 meeting, which starts tomorrow, April 2nd, and runs through Tuesday, April 5th. As a heads up, please note that this event is being recorded and may be shared publicly on EV channels. Many of you are already registered to attend our in-person meeting, and we look forward to seeing you there. If you're not registered yet, or if you would like to get our press materials electronically, you can find out how to do that at either of these 2 URLs. We offer complimentary press passes for qualifying journalists, and our media team is here to help you get registered, connect you with researchers, or generally be a resource to you throughout. Experimental Biology has 5 host societies, and this morning we will be hearing research highlights from each of them. Our host societies are the American Association for Anatomy, American Physiological Society, American Society for Biochemistry and Molecular Biology, American Society for Investigative Pathology, and the American Society for Pharmacology and Experimental Therapeutics. So I know everyone's eager to get to the science, So I'll just say a few quick words about how this event is going to go, and then I'll turn it over to our esteemed researchers. Today, we'll start with a 5-minute presentation from each of our 5 speakers. We'll go in the order listed here, starting with Piyush Patti, a doctoral candidate from the University of Georgia, who will share some exciting research about a live biotherapeutic to deliver a treatment for Parkinson's disease. We'll ask that everyone except the current presenter please mute yourself throughout the event. After all 5 speakers have presented, we'll have a Q&A discussion. You can submit your questions at any time during the presentations or during the Q&A by chatting your question to questions. We'll collect all the questions and try to get through as many as we can before closing out the event at 11:45. So I will now turn it over to Piyush Patti. Piyush is a doctoral candidate in the laboratory of Dr. Anu Kampasamy at the University of Georgia.
Well, good morning, everyone. Well, thank you, EB and the host society, American Society of Pharmacology and Experimental Therapeutics, for this opportunity. Now, microbes play a powerful role in the maintenance of the human body. And today I'm thrilled to share with you how our lab, in collaboration with researchers at Iowa State University, has harnessed the biosynthetic capability of a probiotic bacterium and re-engineered it to produce a therapeutic that might be beneficial for individuals afflicted with Parkinson's disease. Now, before I indulge you with the novel microbiome-based therapeutics, I want to highlight the rationale for utilizing live biotherapeutics as a strategy for the treatment of PD symptoms. Next slide, please. Now, Parkinson's disease is a debilitating progressive neurodegenerative disease that impairs individuals' ability to walk, talk, sleep, and disrupt its overall quality of life. As one of the most prevalent neurodegenerative diseases, more than 60,000 individuals are diagnosed with PD every year, with an expected total to rise to 1.2 million individuals in the US by 2030. This places immense strain on not only the families of the individuals affected, but also the caregivers and the healthcare system. Now, the status quo for symptomatically treating Parkinson's disease is to give oral levodopa tablets. Next slide, please. Now, it's developed back in 1960s. Levodopa to this date is the most efficacious gold standard drug for Parkinson's patients. The mechanism simply works by replacing the brain signaling molecule dopamine, which is severely depleted in Parkinson's patients. Dopamine plays a critical role in preserving proper motor control, as well as non-motor related effects, such as cognition and depression. Now, in order to achieve this sort of symptomatic relief, Parkinson's patients often take 3 to 4 tablets a day. This form of non-continuous pulsatile or wave-like delivery can often lead to a more troublesome side effect, known as levodopa-induced dyskinesia, abbreviated here as LID. As you can see in the video, the individual exhibits erratic, abnormal movements with a complete lack of voluntary control. Now, one hypothesis to the generation of LID is the high fluctuation of levodopa dopamine concentrations caused by this non-continuous high-dose oral tablet delivery and subsequent sensitization of the brain dopamine receptors. Therefore, our goal was clear. Next slide, please. We wanted to create a system that delivers L-Dopa systematically, continuously, in a non-pulsatile manner. For this, we chose microbes as our chassis for therapeutic delivery. Next slide, please. Now, by using modern synthetic biology and microbe genome editing tools, we have designed and developed a stable genome-integrated L-Dopa producing bacterium termed as levodopa live biotherapeutic, or LDLB for short. Using both rodent and canine preclinical models, we have demonstrated that our LDLBs can deliver L-Dopa systematically in a continuous, non-pulsatile manner, all while maintaining concentrations within a therapeutic window. Now, this stable delivery outperformed oral L-Dopa, resulting in a significantly higher dopamine concentrations in the brain. Furthermore, our LDLB is safe and well tolerated in both rodents and canines, and was effective in significantly improving motor and non-motor deficits. As important as it is to develop an LDLB to improve Parkinson's symptoms, the feasibility of translating this project is equally crucial. So with that in mind, we have developed a pipeline for downstream manufacturing for our LDLBs. Therefore, based on our scientific evidence and comprehensive evaluation from our preclinical models, we are optimistic our LDLB will be successful in upcoming non-human primate and human trials. As this field continues to flourish with an already growing number of clinical trials utilizing microbes to reduce disease pathology, our promising new therapeutic is a novel approach to treating people who suffer from PD and beyond. Next slide, please. So we're thankful for the funding for both NIH and National Institute of Neurological Diseases and Stroke and National Institute of Biomedical Imaging and Engineering, as well as the support from endowed research funds from Iowa State University and University of Georgia. As a disclosure, Dr. Kentasamy has an equity in 2 startup companies, PK Biosciences and ProBiome Therapeutics, and our patent for LDLB technologies has been published. We're an extremely collaborative group and are currently hiring tenure-track positions at the Center of Brain Science and Neurological Disorders, where Dr. Kentasamy is the director and chair. You can contact us by scanning our respective QR codes. And with that, I conclude my session. Thank you for your time.
All right, thank you, Piyush. Next, we'll hear from Dr. Catherine Lee, a postdoctoral research scholar at Washington University in St. Louis.
Thank you everyone for being here today. I want to take a minute to acknowledge my collaborators, Eleanor Junkins and Catherine Clancy, as well as note that I can be reached on Twitter at that handle right there. Next slide, please. So the background for this project is that we know that menstruation is a bleeding and clotting event that's responsive to immune, energetic, and inflammatory stressors. And after COVID-19 vaccination, a number of people reported changes to their menstrual bleeding or other menstrual symptoms. But this discussion often centered cis women, that is, women who were assigned female at birth and identify as women as adults, and concerns about fertility. But queer-friendly social spaces, social media spaces quickly mobilized to bring awareness to other demographics. Next slide, please. So my collaborator and I did an online survey that was open for 6 months of last year that garnered over 165,000 responses. But for this presentation, I'm focusing on 552 respondents who reported they usually don't menstruate, use testosterone or gender-affirming hormones, and are not cis women, which is great timing because yesterday was actually Trans Day of Visibility. Next slide, please. Our goals in our survey were to listen to the experiences of people reporting changes so we could ask questions that allow us to look for correlations within our sample to motivate future hypothesis testing. And we wanted to use inclusive language and gather qualitative data as part of this. Next slide, please. So among the people who, the 552 people, we allowed people to select multiple gender identities because that's how a lot of people think about their gender. And you can see that most people identified as transgender, but there are also a number of people who are non-binary or man identified or genderqueer, gender non-conforming. Next slide, please. The results from just our multiple choice question about what types of menstrual symptoms people experienced were that around a third of people experienced some form of breakthrough bleeding after their vaccine. Around 9 and 11% noted that they had chest or breast soreness or hot flashes, which are things that are also associated with this, with having periods and sort of this HPA-HPO axis, but around 37% had no period-like symptoms. So it's not a universal experience. Next slide, please. Importantly, though, is the qualitative responses. With the first one, noting that it was very sudden and upsetting. They heard online that periods could be a side effect, but it causes them dysphoria. And if they had not been warned beforehand, they wouldn't have had supplies because they hadn't had a period in several years. Next. This person reported that it came as a shock after not having a period for 2 years. And they're hoping that their participation could help even one person not go through the dysphoric gut punch, as they phrased it, without knowing about it in advance. Next one, please. And as this person notes, for assigned female at birth trans people, the prospect of period-related side effects has been really scary. And that they know people who are too scared to get vaccinated because they're afraid of experiencing period dysphoria as a result of their vaccine experience. Next slide, please. All of this together highlights that it's more than an inconvenience for trans and gender nonconforming, gender diverse folks to experience a period when they've gone out of their way to eliminate this experience from their lives. We know that it can be a warning sign of multiple forms of cancer, and it can lead to expensive and uncomfortable, worrying diagnostic screening. It can cause feelings of gender dysphoria, and it can be really difficult for a lot of people to find a qualified medical provider who can address and treat them properly and respectfully. Furthermore, it's a surprise that they might not be prepared for with pads or tampons and menstrual cups, and it could increase the risk of interpersonal violence and if they are experiencing their period in public settings and having to address their period among people who are transphobic. Next slide, please. The main limitations of this work is that it was an opt-in snowball sample and it's not representative of the general population. So we can't speak to the prevalence or incidence of these experiences based on our sample. Next slide, please. But these are experiences that were not captured by existing clinical trial or surveillance monitoring systems. And so it's important that we've actually taken the time to listen and acknowledge that these experiences are occurring. Next, please. So in summary, the data exists because we listen to people. Inclusive research design is important. Lack of evidence in clinical trial literature does not mean these changes aren't happening. Changes in menstrual symptoms matter and can worry people even if they're not worried about fertility, and dismissing people's concerns is harmful and seeds mistrust. Next. Thank you. And I will turn it back to everyone else.
Thank you, Dr. Lee. Next, I'm pleased to introduce Ibrahim Musa, who is a doctoral candidate at the New York Medical College, to discuss a compound that shows promise for alleviating food allergies.
Thank you for the introduction, Ms. Johnson. Thank you everyone for attending. Here's my title page. Next slide, please. Food allergies are a highly prevalent disease that impacts nearly 30 million people in the United States, and it's managed primarily by food avoidance. Main form of treatment of food allergy is epinephrine, antihistamine, and corticosteroids. Next slide, please. Fumionectin is an O-methylated isoflavone that is derived from Sophora flavicentis, or red clover, and it's been shown to have anti-mutagenic, anti-inflammatory, anti-carcinogenic, and antioxidant effect, and we believe it potentially has an anti-allergic effect, which we're investigating. Next slide, please. So what are the mechanisms and mediators of food allergy? Upon ingestion of a allergic product like peanut, it gets broken down into other proteins by dendritic cells, which activate T cells, and the T cell deliver K signals to activate the B cells, which produce immunoglobulin E that binds to the mast cells, which upon contact with the allergen protein will degranulate and release the mediators that are responsible for the local symptoms like swelling, itching, and other severe anaphylactic reaction. And as you can see in the figure, we've seen that fumionectin decreases IgA molecule and also binds to mast cells. So therefore, we wanted to investigate the mechanisms by which it does this. Next slide, please. To investigate the mechanisms by which fumionectin decreases production of IgA in B cells, we used a combination of two approaches with computational technology, and we validated the targets that we see using qPCR to measure the gene expression. So for the computational modeling, we identified 516 targets for food allergy, 308 targets for IgA disease, and 332 targets for mast cell diseases. And from database, for compound database, we identified 16 targets of fumonectin in different diseases. So we mined those targets of fumonectin into the allergic disease targets, and we uncovered 25 fumonectin targets in food allergy, 19 fumonectin targets in mast cell diseases, and 51 fumonectin targets in IgE diseases. We took all these targets and analyzed them in geo analysis pathway, KEGG pathway, and constructed a protein-protein interaction network and validated the targets. Next slide, please. On the left right here is a compound target disease network, and on the right is a protein-protein interaction network. The circles are the targets that was uncovered from the computational technology, and the circles that have the orange circles on the left figure are the targets that are very relevant. Examples are NF-kappa B, STAT1, STAT3, TP53, TYK2, and CASP8. And on the figure on the right here is a protein-protein interaction network. The lines are the interaction between the different nodes. These nodes are for the protein targets, and the most significant is the TP53 target, which you see that the biggest and the largest cycle in the center. Next slide, please. So we validated the targets that we see. We took the most relevant targets, and we wanted to see if this computational technology is really identifying the correct targets, and if there is indeed fumonectin is regulating these targets. To do this, we cultured fumonectin with a plasma human B cell line, and after the culture, we cultured for 72 hours. We isolated the RNA, and we use qPCR to measure the gene expression. And from this figure, the blue bars at the untreated culture group, while the purple bar is the group that received the fumonectin treatment. And P53, TYK2 gene, and caspase-8 gene were both, the gene expression were decreased by fumonectin. Next slide, please. And for BCL2 gene, NF-kappa-BIA target, and BTK target, we saw that fumonectin increased the gene expression. Next slide, please. In conclusion, computational technology, using this approach of computational technology and experimental approach is very promising. It helps to save time, it's very good for utilization of resources. We believe fumonectin is going to be a potential candidate for treatment of and prevention of allergic diseases. And our next phase is to do a preclinical in vivo study using a peanut allergic mice model. Next slide, please. And I would like to thank my my PI, Dr. Jimin Lee, and other lab members, Dr. Yang, Dr. Srivastava, and Anish Maski, a PhD candidate. Thank you.
All right, thank you, Ibrahim. Our next speaker is Abigail Kelly. Abigail is a research assistant in the laboratory of Dr. Deepak Panigrahi at Harvard Medical School's Beth Israel Deaconess Medical Center in Boston.
Hi, everyone. I'm excited to present this ongoing project, immunonutritional targeting of cancer via eicosanoids. Next slide, please. So to start, I'm just going to give a brief overview on immune checkpoint inhibition or immunotherapy. So over the last decade, immunotherapy has taken off as a treatment option for many tumor types. And in 2018, the Nobel Prize in Physiology and Medicine was awarded to James Allison and Tasuku Honjo for their critical work on immunotherapy. Immunotherapy won the Nobel Prize because it creates a durable response in patients who previously had no treatment options. The basic premise of immunotherapy is stimulating the body's natural immune defense to various cancer types. Next slide, please. And so, however, despite being remarkably effective in those patients who do respond, immunotherapy does not work in up to 80% of patients and can lead to toxicity and stimulate a pro-inflammatory cytokine storm, which is characteristic of many inflammatory diseases. Next slide, please. So this is relevant to cancer because inflammation has recently emerged as a new hallmark of cancer, as research from many labs has suggested that inflammation can promote tumor growth. Inflammation in the tumor microenvironment can exacerbate tumor progression, as summed up in this quote: "If genetic damage is the match that lights the fire of cancer. Inflammation may provide the fuel that feeds the flames. Next slide, please. So as inflammation is relevant to cancer, our lab, along with others, has been focusing on combining anti-inflammatory treatments with traditional cancer treatments, such as chemotherapy and immunotherapy. Soluble epoxide hydrolase inhibitors are one of the anti-inflammatory drugs that we're focused on and what we used in this project. So I won't go too far into the pathway, but inhibiting SEH stabilizes anti-inflammatory metabolites. And notably, common medications such as aspirin target other parts of the arachidonic acid metabolism pathway. And icosanoids, which are a class of lipid signaling molecules, are derived from arachidonic acid and contribute to inflammation. SEH inhibitors have been shown to improve various inflammatory diseases, like those listed on the right, in preclinical models and are FDA approved for treatment of diabetic neuropathy. Next slide, please. So another compelling target in cancer research is regulating the immune system via dietary interventions. So this is relevant to inflammation, as certain diets are considered pro-inflammatory while others are considered more anti-inflammatory. What we consider the typical Western or American diet is typically high in omega-6 fatty acids, which are generally considered pro-inflammatory. On the other side, the typical Mediterranean diet is high in omega-3 fatty acids, which are anti-inflammatory. And increasing dietary omega-3s has been demonstrated to be beneficial in some cancer studies. Next slide, please. So for this project, we decided to combine dietary intervention with an anti-inflammatory treatment to see if we could improve the efficacy of immunotherapy in preclinical cancer models. So we started mice on either a high omega-3 diet, a high omega-6 diet, or a normal research diet. And then we injected tumor cells and treated with immunotherapy, an SEH inhibitor, or a combination of both. And we did this experiment across 3 tumor types: lung cancer, bladder cancer, and skin cancer. Next slide, please. So we found that a high omega-3 diet enhances immunotherapy in the bladder cancer and lung cancer models. So these tumor curves that show tumor volume on the y-axis and the treatment days on the x-axis show the difference between mice that were treated with immunotherapy on the control diet versus on the omega-3 diet. And these results suggest that a high omega-3 diet could be beneficial for patients receiving immunotherapy. Next slide, please. So interestingly, we also found that the combination of immunotherapy and the anti-inflammatory SEH inhibitor worked best in mice on an omega-6 rich diet. In the bladder cancer model, the SEH inhibitor synergized with immunotherapy to dramatically reduce tumor growth compared to either therapy alone. So in these experiments, we start with large tumors, and each drug alone, you can see it doesn't work. It's similar to the control tumors. But when you combine them, there's this potent anti-tumor effect. We also observed this trend in the melanoma model. And these results are encouraging, especially because the omega-6 rich diet is representative of the typical Western diet, implying that the combination of an SEH inhibitor with immunotherapy could be clinically relevant. Next slide, please. So based on these preclinical studies, dietary supplementation with omega-3 fatty acids could improve the efficacy of immunotherapy treatment. Additionally, immunotherapy and SEH inhibition exhibit synergistic anti-tumor activity in mice on high omega-6 diets. And so these results are promising, and the combination of immunotherapy, SEH inhibition, and dietary supplementation should be validated in human patients. Next slide, please. So just a big thank you to everyone in the lab for their continued support, and to our collaborators in the HAMMOck lab who are heavily involved in the design and synthesis of SEH inhibitors. Thank you.
All right, thank you, Abigail. Our last speaker this morning is Dr. Aileen D'Souza, who is a postdoctoral fellow at Georgetown University. Dr. D'Souza will share her research on possible long-term health impacts of caloric restriction.
So good morning, everybody, and thank you for the invitation. So I'm going to talk about the weight cycling and the consequence to the heart in the future. So next slide, please. So the weight cycling can be done by voluntary decision or non-voluntary. For the voluntary way, we have the overweight perception situations in athletes or performers. For non-voluntary, we have situations of food insecurity that is present in situations of war, food shortage, or poverty. But in both situations, we have a very low food intake that leads to a weight loss. After some time, people, they have access to the food, or they decide to eat normal again, and they have the weight gain. But then a new cycle starts and a very low food intake, low weight loss. So this is a weight cycling situation. So next slide, please. Here's one example for the voluntary weight cycling. That is the low calorie diets. And this table is showing the prevalence of the number of people doing very low calorie diets over the years. And you can see it's increasing a lot, either for men or women. Next, please. And another example, but for the non-voluntary decision for the weight cycling, it's situations of wars and pandemics. But what is the physiological consequence of this weight cycling? Next, please. So to understand a little bit more about that, we used female Fisher rats and we divided in two groups. The first group is the control group who had, this group had free access to the food all the time. The second group, we did food restriction in 6% for 2 weeks. After 2 weeks, we gave free access to the food for 3 weeks. And we repeated this cycle of food restriction and ad libitum diet for 3 times. Next slide, please. So on your left side, you have the food intake, in blue for the control and pink for the yo-yo diet. And you can see every time we did the food restriction, the food intake was dropped, but they quickly recovered to the normal eating pattern. And at the end, on the purple arrow, you can see they were matching to the control levels. On the right side, you have the body weight, and you can see the body weight was following the food intake reduction, and they lost the same amount of weight for each cycle. Next, please. So the next experiment, we need to measure the cardiac function, and we use the ultrasound. Here's just an example for the image where each wave is showing the heart contraction and relaxation, meaning the diastole and the systole for the heart. Next, please. And using that technology, we could measure the cardiac output, that is basically the heart rate multiplied by the stroke volume. For the cardiac output, we saw the yo-yo diet, they had a lower cardiac output after the 3 cycles. The heart rate, we didn't see any difference, but we have a tendency to have a lower heart rate, and the stroke volume as well. Next, please. We also measured the insulin and glucose levels on those animals at the end of the 3rd cycle. So the glucose, we didn't see any difference for the fasting period. And the insulin, we saw the animals under the yo-yo diet, they had a higher insulin levels. And when we compare the ratio insulin glucose, that is the HOMA index that is used in clinic to see the insulin resistance, we saw the yo-yo diet at the end of the 3rd cycle, they had a higher HOMA index, meaning they can have a higher insulin resistance. Next, please. So basically, we can say the weight cycling is causing fluctuations in the blood pressure and the heart rate every time you have a cycle, and also fluctuations in glucose levels. And both situations can lead to increased cardiac load and vascular injury. And at the end, in the future, that can lead to a cardiovascular and renal disease. Next, please. In conclusion, despite the normalization of the body weight after 3 cycles of yo-yo diet, the animals, they had reduced the heart function and higher insulin resistance. Next, please. And we can say that individuals who have experienced cycles of inadequate food intake from different causes might be at increased risk of developing cardiovascular disease and diabetes in the future. Next, please. And based on that, I want to say thank you to all my collaborators and Dr. Sandberg Laboratory, Dr. Alaniz, Echo Bagger, Kyle, and Dr. Rodriguez. Thank you.
All right, thank you, Dr. D'Souza, and thanks again to the rest of our presenters. Now we will transition over to a Q&A discussion. Once again, I'll ask that everyone except the current speaker, please keep yourself muted. We will read the questions submitted via the chat and invite our speakers to comment. We've gotten a couple in so far, and yeah, please just chat your questions to the questions recipient, and we'll read those aloud. So first up, we have a question for Dr. Lee. Can you please, what method was used to determine statistical relevance in choosing the 552 patients in the sample?
So I'm not sure what's meant by statistical relevance. This is more a descriptive project at this point for this sample. We're doing follow-up data collection as well. So for the moment, what I'm presenting at this conference is just the initial description of people reporting this side effect, because it's been not covered sufficiently, despite being noted by lots of people.
Thank you, Dr. Lee. Next question up is for Dr. D'Souza. Would you expect age to make a difference? Could these sorts of fluctuations have different long-term impacts if they happen during childhood, adulthood, or in old age?
Yes, because especially for young age, you are under the development, so that can be compromised. So I expect worse outcome. Aging, maybe the diet can be more aggressive. So yeah, so I believe it different effects.
Okay. Next question is for Ibrahim Musa. Are there particular types of food allergies that you would expect this compound to work better or worse for?
We would not be able to tell that at this stage of the investigation. This is just an in vitro research, but most likely based on the severity of food allergy, with peanut being the most severe and going down gradually to, let's say, wheat and soy. This compound will, our in vitro studies, we use, in vivo study, we will use peanut as an allergic model. So if it works perfectly with peanut, then it's most likely going to work out for the remaining food allergies very well.
Okay, great, thank you. This next one is for Abigail Kelly. Are there any downsides to omega-3 supplementation for people with cancer, to your knowledge?
That's a great question. To my knowledge, the results on omega-3s in human patients for cancer have kind of been hit or miss, some showing beneficial effect, but no negative effects to my knowledge.
All right, for Piyush Patty, what happens to the bacteria in the long run? Will they stay in the gut and keep synthesizing L-DOPA or die off after a certain amount of time?
Yeah, that's a great question. We have done extensive what we call colonization studies, and we see that the microbe doesn't actually colonize in the gut, and it gets rapidly excreted by within 48 hours. The peak time. So we expect that our dosing paradigm would be, we're giving it every 12 hours right now, but by further optimization and further genetic editing, we can hopefully colonize it up to 24 hours.
Okay, and while we have you on the line, another one, Piyush, about your study, what might be the, how do you envision the biotherapeutic, or how do you ensure the biotherapeutic doesn't become overactive and produce too much L-DOPA?
That's a great question. And designing the microbe in a sense that we have, like I mentioned, is a genome integrated system. And we have specific promoters that are designed, and we've optimized those promoters, which then allows the production of our therapeutic, which is L-DOPA. And we can control the production by giving different concentrations of the inducer, which then causes this production. So by not just controlling the genetic production, we can also control therapeutic level by classic dosing paradigm. So we can give various concentrations of ranging from 10 to the 8 cells to 10 to the 11 cells. And we have done this studies in our preclinical models, and we have demonstrated that L-DOPA production can be modulated basically from upstream bio manufacturing processes.
Okay, thanks. Another one for Dr. Lee. What would you say to someone who is hesitant to get a vaccine based on the results of this study?
I would reassure them that the vaccine is safe, it is effective, it lots of people are not experiencing any of these reactions at all. And the effects of getting COVID, especially if you get COVID and you were not vaccinated, are much, much worse, like long-term systemic disruption of lots of systems. So I would still highly encourage, I just think it's nice in the same way that we tell people, you know, hey, you might have a fever and fatigue for a day or two after the vaccine, for people to be prepared for this to be potentially a side effect.
Okay. Looks like we just have one more question in the queue. So if anybody has any other questions, please go ahead and chat them to questions and we'll read them aloud. The last question I have in the queue so far is also for Dr. Lee. What might be the mechanism by which the vaccines affect menstruation to the extent that this can even overcome the effects of testosterone?
That's a great question. So, and something that we have applied for funding to look into more specifically. What we suspect is happening is that the immune response of the vaccine, because it's such an immunogenic vaccine compared to, say, what we usually get for the flu shot, is kicking off this big immune response, which leads into all of these systemic inflammatory processes. That's why we get these generalized systemic inflammation effects like the fever and the fatigue, in addition to the localized ones like soreness. And what I suspect is happening is that because the uterine lining is already sort of regulated by pro and anti-inflammatory mechanisms that are controlling the building and sloughing, that there's just a little bit of crosstalk that happens between the systemic inflammation and the localized pro and anti-inflammatory processes in some people, and that is sort of disrupting this regulatory process.
Okay, thank you. We got in another question for Piyush. Do you think this engineered probiotic could be used as a prophylactic measure?
Yes, that's again, these are great questions. And I think that, yes, it can be used as a prophylactic measure. Our target for giving this probiotic is in the early stages of PD. We are expecting that when Parkinson's patients with diagnosis, currently how it is, we expect to give our biotherapeutic in the early stages of PD. For late stages of PD, there's more aggressive approaches that are still currently under testing, but our goal is for early stages of PD.
Okay, great. I'm not seeing any other questions coming in on the queue. Oh, one just popped up. This one is for Ibrahim. Are there any existing drugs that suppress immunoglobulin antibody production? If so, do you think this compound could potentially be complementary with other medications or work better in some way?
That's an excellent question. There are other drugs that do decreases immunoglobulin production, but in overall, we are not trying to decrease the anti-antibody production because you also want to keep the normal protection that the person has. But for this compound, we know that if this compound is very, very good with the B cells, and it decreases IgA production from the B cells. Unlike the other compounds that is available for treatment of IgE, which is binding to the IgA molecule itself, this compound decreases the production of the immunoglobulin. So if you combine it with a different compound that is being used already, it will potentially be very beneficial for the patient. Thank you.
Okay, thank you. And Dr. D'Souza, your study involved cycles where caloric intake goes down and then up again. Would you expect the results to be different if caloric intake goes down and stays down?
Yes, it's going to be more aggressive to the body. And I think the outcome is going to be worse in this case, if it keeps down. Because you don't have enough protein for the body, energy, everything. So the heart is going to get more damage. It's a good question. We have to try, but I guess it's that.
Okay, thank you. And Abigail Kelly, are there any clinical trials studying omega-3 supplementation during cancer treatment, to your knowledge?
Yes, I believe that some have been conducted. That's what I was referring to before with the mixed results.
Okay, great. I think that is all the questions that we had coming in. So, and we're right at time. So I will go ahead and wrap it up. Once again, I want to thank all of our speakers for sharing their work and all of you for joining us today. Here again are the links for the meeting and our virtual newsroom. The meeting starts bright and early tomorrow morning, and we hope to see many of you there. At the virtual newsroom, you can find press releases for all of the research covered today, as well as many other exciting research highlights from the meeting. So please check it out and let us know if you have any questions. Thank you again, and I hope you all enjoy the rest of your day.